So what I'd like to do, I guess, is give you a little bit of an orientation, and the home page is always a good place to start to see sort of how we interact with the data in Phytozome. And then we'll just go off and do the sort of explorations you would do if you were interested in a particular set of genes or set of genomes and wanted to explore them a little bit interactively, and then possibly grab the data and take it offline to work with somewhere else. So to begin, this is the home page of Phytozome, and kind of the first thing you want to be able to answer for yourself is what genomes, what organisms are included in Phytozome. And there are a couple of ways you can find that out. If you're just interested in what's come out recently in Phytozome, there's a list at the top of the overview, which is going as far back as about end of 2019 to our most recent releases in March. So that shows the genomes that have been coming through, and each one of these is linked to an information page for that genome. I'll just quickly click over to one of them and you can see what's in there. It's typically an overview, some statistics about the genome, a description of how the assembly and annotation were done, and then possible restrictions on data set usage. I'll get into that a little bit later.
Hey, David, could you zoom in your screen just a little bit bigger? Is that possible?
Is that good or more?
That's better for sure. Thank you. Yeah, that looks good. That last
Okay.
one.
All right, I'll keep it there. But that's only a subset of the data. Another place where you can see all the genomes that are in Phytozome are arranged in this, what we call a target tree, which is a way of selecting genomes in Phytozome. And this is not a strictly formally calculated phylogeny. This is basically following NCBI's organization with a lot of collapsing of internal nodes. So please don't base your research out of the arrangement of the tree. It's just a simpler way for us to present the data. So going through this tree, you could pick a genome either by simply clicking on it, then all your subsequent actions will be applied to that genome. You can actually search for particular genomes up here, and when you select them, then that genome will be selected. And you'll always have a list here of which genomes you've selected. Could I ask, is the Slack panel obscuring anything, or you can fully see this on the right-hand side?
You can see it.
Yeah, we can see it. It's good.
All right. So that's a list of all the genomes I've selected. And again, I can get to their information pages by just clicking on the links here or clicking on this information link. Two more ways to get to all the genomes in Phytozome. If you don't like phylogenetic organization, can never go wrong with alphabetical. You can probably find the genome you're interested in here as long as you know the genus. This will take you, you stop at one of these, this will take you to the information page for that genome. And then lastly, in the release notes is some information about all the genomes here, where they were assembled and annotated, what versions they're at, common names if they're available, whether there are any restrictions on the data usage, and a measure of the annotation quality. This is the Busco scores against a sort of pan-eukaryotic gene set and a pan-embryophyte gene set. Obviously, the closer to 100%, the more confidence you have that core eukaryotic and embryophyte genes have not been left out of the annotations. You can, as in almost all of the tables in Phytozome, you can always export these to work with them later. that can be handy if you're preparing a publication or just want to keep track of the genomes you're working on. It'll include the links back into Phytozome. And I'm not gonna, yeah. You can just see the sort of data that comes out, the link to the organism info page, assembly and annotation site, data usage policy, and the BUSCO scores that I mentioned. Okay, so a little bit more about what information is being exposed here in the target tree. You can see these little colored lozenges here that tell you right off the bat whether the data corresponding to these genomes is public and unrestricted, or whether there are still some restrictions on it. There's also an indication of whether the genome was assembled and annotated externally at another site besides JGI, or whether it was done at the JGI. And then lastly, there's information about whether this genome is also part of a special project on Phytozome, such as the Gene Atlas project here for, well, I want to say Fiscometrella, but now it's Fiscometrium patents, or the Open Green Genome project, which has this Ceratopterus genome included in it. All right, so that's all the data that's in Phytozome, and depending on what you want to do with it, this is a good place to start. Let's say you're interested just in grabbing bulk data files, assembly FASTAs, gene sets, GFFs. You're going to do work with them offline. You could select those. I've selected 3 genomes, and then just say that I want to get whatever standard data files are available for those. So I would just click on that. That would take me off to the JGI data portal with those 3 genomes selected. I'll expand them out. And you can see the type of pre-compiled bulk data that's available. That can include the assemblies, hard masked and soft masked assemblies, annotation information, FASTA files of the transcripts, of the CDS, of the proteins, GFF files giving you the gene structure, or the repeat masking features, and then typically a README and a data release policy file. You can select these and download them and do as you wish with them offline. It's probably a good time to mention that data download always requires that you be logged in, which means you have to be registered at the JGI and logged in, and you'll see in a little bit what happens if you're not, since I'm not going to log in right away. Is everyone already registered at the JGI? If you're not, we may take a short break before going into some of the examples to make sure you can register and get a.
David, I bet some people aren't registered. If you have questions about that, ping me on the chat. Happy to help.
Okay. A little bit more about what's accessible from the home page. Looking at the top menu, this is a link into just the overall JGI site, so not specific to Phytozome or plants. I'm sure you've all visited this at one time or another. If you wanted to go directly to that data portal we were just at, but make your organism selections there, this then links into the data portal for all the genomes in Phytozome, and then you can select them yourself here. I find it more convenient to make my selections on the Phytozome homepage and then go to the data portal. We have a number of tools that you'd expect on any genomics website. There's a BLAST interface we'll get to a little bit later, a keyword search, which is in fact just what you see here on the homepage. There are genome browsers. Our genome browser is adapted from JBrowse from the Gmod project. We have a data warehouse biomart that has pretty much all the data we've ever generated in it, and it's typically quite easy to work with. Phytomine is another data warehouse that is not as complete as Biomart, so we do the updates to that a bit less frequently because it's a bit more involved updating it. So it doesn't always have the latest genomes that we've worked with. And then we also have a Synteny tool, which I'll show in some of our examples, which is interesting to work with. Projects, I'm not going to talk about too much right at the start. We'll mention that a little bit later, but because there's so much data in Phytozome right now, there are 274 genomes. A lot of times you would like to reduce the size of the set you're working in, so you're not overwhelmed when you look at BLAST results or gene homology and such. And the projects provide a nice way to do that. They also provide an opportunity to have targeted news and targeted information in the overviews that aren't applicable for the general Phytozome project. I showed you the genome section for getting to a particular genome right away. The cart, which will not be happy because I'm not logged in. So the cart is a typical data cart where you can add genes, create lists, share them with other people, download them, etc. And we'll work with that a little bit later. The contact is just the usual JGI, the usual Phytozome email, phytozome@jgi-psf.org. That is the best way to contact us. A good 30%, 40%, sometimes 50% of the group suffers from insomnia. So pretty much anytime you write there, you will get a response very quickly, and we're happy to help. The subscribe is for a group mailing list that is posted to very infrequently. It's usually about planned maintenances when the system will be unavailable. Again, we saw the login, and this last bit here, I will just tease, and we'll work with it a little bit more later. But this is a way for you to not just rely on those different projects to reduce the number of genomes you're looking at, but you can explicitly decide which genomes you do or do not want included. For example, I might be studying the grasses, and I might not, however, be interested in all 57 of the Brachypodium distactyon lines from the Brachypan genome project. So I might just decide to hide that set. I've just clicked on the name, and those are hidden. And now if you look at the target trees, and we'll see it also when we go into results, those are no longer there. The Brachypan genome set is gone. So that can be convenient to work with. It's easy to forget that you've hidden certain genomes. So there is probably a bit too small little warning bell that appears over this gear icon saying that you're excluding a certain number of genomes. You can always undo it by just going all the way to the root of the tree and selecting everything again. And now we're back to where we were. Okay. Let's actually probably know enough already that you can start looking for things and you're finding and find them. So let me can probably put this in the chat. Actually, I can just share it up here since I'm showing the whole screen. So if I wanted to find all the genes, all the TIC, all the time for coffee, circadian genes in the set of genomes that are basically the super astrids and the super rosids. So that is this set. Here are all the astrid genes below here. Here are all the rosid genes below here, and I want to search against them. How would I go about doing that? Well, I'd select the set on the tree. Okay, and you'll see that once I do that, these action buttons become active. When I have nothing selected, I can't actually do anything. But as soon as I select some genomes, I can. And the full set of genomes I'm selected is in this list here, but you can also see it on the tree. Okay. So I'm going to now search that set of genomes, those 135 genomes, for time for coffee genes. And then what I quickly get is all the genes that match that phrase. And if there is more than one gene from a particular organism, those search results come back collapsed. But if there's only one from that organism, then you'll see it displayed directly there. So the search results tell you what organism it's from, the gene identifier, the def line or description. All of these descriptions that start with a parenthetical X of Y, certain number out of a total set, those are automatically generated def lines. Those are not assigned by manual curator. And this just means that there are 4 other genes. Well, you can even see it here. There are 4 other genes in this genome that were assigned the same def line. So as I mentioned, whenever I have a table in Phytozome, if I just want to work with the results offline, I can always export it. And I'll get links to get back to the particular genes, their description lines, all the information I see in the table. So that's handy for working offline. If I wanted to actually do a little bit more specifically with a set of genes within Phytozome, what I might do is save this set to the cart. And unfortunately, in this example, there is not a select all when there are multiple collapsed results. So I am just going to grab some of these for now. And I want to add them to the cart. I have some of them selected, but I'm still not able to do that. And that's because working with a cart is something that requires you to be registered and logged in. So I'm going to sign in, and I'm probably going to lose all my selections. So let me just grab a few of them again. But now you can see that the add to cart button is active. So I can add these to the cart. It noticed that I have some genes already in the cart. It'll ask me if I want to add these or overwrite the current cart. For now, I'm going to just overwrite it. And now those genes are in the cart. I'm just taking a look at them here. Once they're in the cart, I can do things like save them to lists, share the lists with people, download information about those gene sequences or annotation information. So it's a handy tool. The other thing you can do with a search result is these are all persistent. So you can take this URL and share it with anyone else. So if you want to tell them a particular set of genes you're interested in or that you're working on, you want their opinion of, you can actually just send them that URL and they'll be able to pull up exactly the same list. Okay, are there any questions about that? We haven't drilled down on what you can actually learn about these genes yet, but this is a way to find them and collect them. Okay, so that's a general search with looking for a particular def line. You can also, of course, search directly with gene identifiers. So if I just asked you to find a particular Arabi gene and I knew the locus identifier, what do? Well, I could drill down here and specifically select Arabidopsis, but I know that the Arabi identifiers are fairly unique to Arabidopsis, so that I can really just select all the genomes. And I know I'm not gonna find a lot besides just the Arabi genome. So let me search for that gene. Okay. And you see I got 2 results, and that's because we have 2 versions of the Arabidopsis thaliana annotation. One is the last one produced by Ter, which is version 10. And then there's a version on the same assembly, but annotated with additional RNA-seq data. That's version 11 from the Airport group. So both of those are in Phytozome. You can decide which one you want to work with. And then lastly, if I have unique identifiers like Unipro identifiers, I can also find genes directly that way. And I probably typed it wrong. Yes, that is not an S. That is a 6. Okay, and the same 2 genes come back because that Unipro ID is associated with the proteins produced by those genes. Okay, are there any questions so far? So I want to actually go through an example, okay? And that is, typically what do we use these comparative websites for is we have interest in a particular organism and its genome, and it's probably not a model system, or if it's a model system, it's certainly a harder model system to work with than the old standbys like Arabidopsis. So what we're usually going to do is try to find the relevant genes in Arabidopsis and then start looking for the associated genes in the other organism. So the example I want to work with now is trying to find some of the analogs of the evening complex genes in Arabidopsis, trying to find them in the legumes and soybean and Phasolus specifically. So the evening complex is 3 genes. It's a transcription repressor complex that's involved in circadian rhythms. So very, very important in telling the plant when to flower, how to shift its metabolism in response to changes in photoperiod, etc. So a lot of interest in it. So what we're going to do is sort of find the relevant genes in Arabidopsis and then see what we can learn about what we think are the equivalent genes in soybean and Phasolus. So there's probably no reason that most of you have this at the tip of your tongues, but the three Arabidopsis genes in the evening complex are called LUX, ELF3, and ELF4, and the ELF stands for early flowering. And the LUX, as you can imagine, is related to sensing light and sensing day length. So what I'm going to do is I'm going to start off working with my old friend, the TER10 annotation of Arabidopsis, which is right here. Select that. And I'm going to look for LUX genes. Okay. And I mean, I encourage everyone, you've got your browser open, to go along and do the same. And you can see that I get a bunch of hits, 12 in this case. One of them actually has the gene symbol of LUX assigned, and I happen to know that that's the gene I'm interested in. But other genes will come up too, just because, again, this is a keyword search. So I mean, we can actually look at some of these genes. Let's look at one of these that's doesn't have the gene symbol LUX, the third one here. And you can see that it's getting caught just because there's this LUX S peptidase domain in it. And that's why it came up in my search, but it's certainly not the one I'm interested in. So let's go back here. I'm going to want to work with the actual LUX gene from Arabidopsis. So I have two links for each gene I find in the search. One is to go to its gene report, and the other is to actually view it in a genome browser to see it in its genomic context. Right now, I want to go to the gene report because that's where a lot of the information related to orthology to other organisms is most easily explored. So let me click through on that and tell you what we're seeing here. So this overview information at the top, we have for every genome. Sometimes there's more, sometimes there's less, depending on which fields are populated. So this is telling me which genome and version I'm actually looking at. So this is the TER10 assembly and annotation of Arabidopsis thaliana. The NCBI tax ID for Arabidopsis thaliana columbia. This is an internal ID that Phytozome uses. We didn't used to expose this, but we decided to because for debugging things, it actually makes conversations go much faster. Phytozome has a lot of organisms that have been annotated multiple times in multiple versions. And so when someone gives us this information, we don't have to have an email thread saying, I'm seeing something strange in poplar. And then we say, which poplar? And then they say, poplar trichocarpa. And we say, which poplar trichocarpa? And so back and forth and back and forth. You give us this number, we know right away what you're talking about. This is the gene ID and also an internal ID for the gene. The advantage of for similar reason, we expose the internal ID because this is unique across all the genomes in Phytozome. This might appear in different versions of the Arabidopsis thaliana annotation. There's nothing prevents someone from using it as an identifier for a gene in a totally unrelated species, though they shouldn't. But this is always unique within Phytozome. So again, we know exactly which gene you're talking about when you need to pass that. So this is a gene locus, and this actually has 3 alternative isoforms at it, which you can see in the list of transcripts. The one that is in bold is the primary transcript. That's designated as the representative transcript at that locus. There is no hard and fast rule for what becomes the primary transcript. That could be set by whoever is working with that genome. It can be based on which is the longest coding sequence transcript at that locus. It can be the one that's expressed at the highest level over a particular set of conditions. It could be the locus that was first explored in the lab with mutants. So don't be surprised if sometimes, like in this case, the primary transcript has a 0.3 suffix as opposed to a 0.1 suffix. The fact that this is surrounded by an orange box means that for this current view of the gene report, that's the selected transcript. What does that mean? We show a bunch of domains and functional annotations for the gene, and those are based on the protein sequence almost always. And different transcripts can have different protein sequences, which means the domains that are found and the annotations that apply can be different depending on which transcript is selected. So it's important when viewing this to know which one we're doing. We're looking at the primary transcript now. The same applies. Let me get down here to the sequences. We shade the sequences to indicate where the UTRs and where the coding exons are. And if I choose a different transcript, the regions that are shaded will be different if the exon structure is different. So always important to know which one you're talking about. I can shift to another one, and you'll see that a certain set of the annotations, the Go assignments, are grayed out. That means they apply to the gene, but not to this transcript in particular. You can also see that UTR is starting a bit further in on the gene locus than it did for the primary. Okay, let me just go back to the primary. Location information where it's found in the genome, chromosome 3 over this range on the plus strand. Some gene symbols or synonyms that have been applied to this gene. As we said, it's the Lux gene. Links out to the UniPro identifiers for the different proteins. Because the proteins can be different, that means the UniPro identifier can be different depending on which transcript you're talking about. This tells me right away, since I see the 0.1 and the 0.2 transcripts together, those have the same protein sequence, and that's why they're getting the same UniPro assigned to them. But the primary transcript has a different protein sequence. It has its own UniPro. A description. If an external genome is given to us and it's actually been annotated with def lines, as of course Arabidopsis has been, there is a non-automatic, a manually curated description associated with this gene, which is a homeodomain-like superprotein. We still do our game of applying a def line automatically to these, and we show them here. And those def lines are basically built by taking things like panthers and PFAMs and KEGG identifiers and descriptions and trying to find a combination of those that's as unique as possible throughout the genome. So in this case, this particular combination of panther families is only found in 3 other genes in Arabidopsis. So that was assigned as the def line. But certainly for a manually annotated genome like Arabidopsis, you're going to typically be handed a def line that's been curated. Links out to particular views. This is a view of this genome in the browser. And let me take you there right now. I'll zoom out a little bit. Let me expand some of these tracks so they're easier to look at. Okay, so this range that's highlighted in yellow, that's the gene locus. You can see the 3 different transcripts. The orange indicates the coding sequence, and the gray is UTR. So you can guess by looking at this that the 0.1 and 0.2 alternative transcripts, they seem to have the same coding sequence. That's why the proteins are the same, whereas the primary has this additional downstream bit of coding sequence on the terminal exon. You can see homology of other proteins to the gene in question on the browser. Don't be surprised, these should only extend over the coding parts because these are protein alignments against the genome. So they're not going to know anything about the untranslated regions. For some of our genomes, like Arabidopsis, we also have gene level expression from the Gene Atlas project there at Hudson Alpha. Thought I saw Avinash on the Zoom a little while ago, but I'm sure he's in the room. He did the lion's share of that work. And basically, these are just colored to show the degree of variation across the conditions of the gene expression. So this is kind of a darker blue, which means that I can expect that there's not a huge amount of variation across the genome, across the conditions in the expression experiment. You can see the expression in these, under these different conditions in leaf and root is relatively comparable. I don't know if I have any handy that are more variable. Yeah, here's a gene that's certainly preferentially expressed in root. So let me just come back to here. All right, so let me go back to the gene page I was at. There's a link here, AT, which is to the Arabidopsis tear site. So you can also view this gene, and it's an original original project site. This S is something we only have for Arabidopsis, and we only have a single data set right now. This is a Arabidopsis transcriptome single cell atlas viewer, and it's somewhat less interesting, sorry, Joe, when you actually pull out, no, this is a better example. A lot of times, some of these genes may only be expressed in a few cells at a few times. But this is showing you a graphical view of where this gene is expressed, and you can actually drill in and check what type of cell at what point in its life cycle. Let me see if I can, yeah. So this is showing only those cells in which this gene had significant expression, and these are color-coded by origin. But again, we only have a single data set. We only have this for Arabidopsis at this point, so it's not something we work with in general, since it's only for this genome. Okay, functional annotation. So these are the results of applying interbro scan to this particular protein. So domains are called out, and family matches from Panther and Gene3D and TigerFam and PFAM and all, and their locations on the protein are indicated. And you can get information about what they're from. All of the all of the Lux genes in all organisms are characterized by this MYB domain, which I think stands for like myoblastic. I think this was first identified as an oncogene in some avian leukemia. That's how that domain picked up its name. Anyway, in addition to the graphical view of those annotations, there's a tabular view. Again, this can be exported, so you have a record of it. But here's where orthology and all gets interesting. So down here are a set of protein homologs calculated by BLAST-P with a little bit of massaging on it downstream. So this shows all the genes in this organism and other organisms that have close similarity to this particular gene. And what I especially want to draw your attention to is this column, Ortho. These are orthology type assignments between the gene in question, this Arabidopsis gene, and the gene being displayed in the table. 1-to-1 means there's a 1-to-1 orthology between them. 1-to-M means this one Arabidopsis gene has correspondence in the other organisms that are multiple. It doesn't have just a single homolog in the other organism, it has multiple ones. Those can be due to tandem duplication, those can be due to whole genome duplications in the other organism. And then in principle, you could get all the way down to M-to-M, which means the multiple copies in Arabidopsis are associated with multiple copies in this other organism and all types. So how to work with this table to find out what you want. So we were interested in the analogs of this gene in soybean. So there are a couple ways I can do it. One way is I can just start filtering this column. I know it's gene max. I have a ton of different soybean strains. And so I want to stick with my canonical reference, which is the Williams 82. And I want to stick with, in fact, the most recent version, which is A4. So I'm just filtering based on that. And so now I've restricted down to just the soybean Williams 82 version 4 assembly genes. And so you can see that right away I've identified 2 soybean genes that are in a 1-to-M, in this case M is 2, 1-to-2 relationship to the Arabidopsis Lux genome. And not surprisingly, they have this MYB DNA binding domain, just like the Arabidopsis Lux has. So 2 things I want to draw to your attention. You can see from this multiplicity number here, there are more than 560 other genes in soybean that also have this domain. So we are not going to find the ortholog for Lux just by searching for genes with that domain. That's why doing a true orthology comparison, like is used to calculate these type assignments, is necessary and useful. The other thing I want to point out is score is what matters, not similarity. You can see that there are higher similarity hits, but with appreciably lower score. What this means is there's a small region of the target gene that has high similarity to a small region of the gene in question, of the Arabidopsis gene. The similarity is only calculated over the part of the proteins that are in alignment, so that can be deceptive. bigger score is what matters. So anyway, two soybean genes correspond to this Lux gene. Let me also then look for the Phasolus gene, the common bean gene. I'll take one of these. It doesn't really matter which one of these soybean homologs, and swing over to its gene page. Very similar annotation to what we saw. And then now I'm going to go here, and I was just going to filter this to find the Phasolus partners, but I'm going to show you the other way of doing that, which was to take advantage of this clade cut capability. So Phasolus is down over here. Actually, what I've just done is I've removed Phasolus. But in fact, I just want to see Phasolus. So I'm going to go up here and I'm going to complement this set, which means I'm going to now turn everything off except Phasolus. And I don't know if you saw while that happened, the protein homolog table updated, and now I only have the Phasolus genes. So you can see the top hit is this Phasolus gene in an M-to-1 relationship, meaning there are more than one soybean genes that hit this Phasolus gene. And we kind of expect that because soybean has had a whole genome duplication since its divergence from the common ancestor with Phasolus. So assuming that those genes had been retained, we would expect more soybean copies than Phasolus copies. Where I'm gonna look to verify this also is I am going to look at a plant family that has a very high similarity to this soybean gene. I'm going to look at its membership. Okay. And these families were calculated with the previous version of soybean. It's not A4, it's A2, but the conclusion is the same. So this particular Phavid family, a single Phasolus vulgaris gene, two soybean genes from the duplication, chickpea just one, red clover just one, as you'd expect, given the duplication history. And then while I'm here, let me add that guide to the cart. You can do the same. Let me go back. I'm sorry. I should have done that when I was here. And add those soybean genes as well. These two, just keep those in the cart together. Okay, so I found these analogs of Lux. Looks like there's one Phasolus and two soybean corresponding to the single representative in Arabidopsis. I want to actually check that a little bit more. Does that really look like that's due to a whole genome duplication, or is that just some silly tandem or segmental duplication that might have occurred? So that's why I'm going to go now to the synteny viewer. from the tools menu. And I am going to choose my Phasolus as one of my genomes, and my soybean, Williams 82A4 is the other. And up pops your usual dot plot. So you can see sort of shadows indicating a whole genome duplication, but you can also see that there's been a lot of rearrangement since then. But let's check the gene that we're specifically interested in. So let me take that Phasolus gene. Oh dear, I didn't use the tab. My bad. It's probably gonna make me reset it. Yeah, sorry. I meant to do that on another tab. Okay, so I copied the Phasolus gene and I'm going to search for it here. Okay, and it says it found it in 2 locations. Let's actually see what those locations are. So it found that gene on soybean chromosome 12 and on soybean chromosome 11. So I see the bit on 11 here, that's highlighted in green. Can everyone sort of see that? I will zoom in a little bit. Right, here it is. So Phasolus chromosome 11. There's a syntenic block with soybean chromosome 11. That's here in green. And there's another syntenic block with soybean chromosome 12. Okay, so those 2 are selected. Let me actually grab a little bit more of the area around there. And so I will select a region. Now everything is selected that I just covered, and then ask for an overview of what the syntenic segments look like. Okay, so each of these different colors is a different syntenic segment from the dot plot, and how it shows up reflected in the partner organism. So Phasolus chromosome 11 is on top. That's Phasolus chromosome 10, and here are the soybean chromosomes at the bottom. Because the gene ID is in the search box, you can see that it's going to highlight the gene pair in Phasolus and soybean, and that is in fact what we have here. You can see that a single gene in Phasolus is getting reflected in 2 different positions in soybean on chromosome 11 and chromosome 12. I can actually look in even more detail, and I'm using a trackpad, so this is always hard for me. Select a region, go to the detailed alignment, and there we are. And let me put in that soybean gene's name, that Phasolus gene's name again. Okay, and you can see from this plot that the overwhelming majority of genes in this region are reflected in pairs on chromosome 12 and chromosome 11 in soybean, and a very large fraction of them have been retained. I can now grab this whole syntenic region And show it in the Finder, and then where are we? The reason I'm trying to find it in the Finder is because when it opens up in numbers, it gets. Oh, okay, good. It is coming up with the headers properly. Okay, so these are the genes throughout that entire synthetic region. I can now analyze these independently, download data about them, whatever I want. I can also, if I'm making publications, I can save an SVG of these images. So certainly based on what we saw with the assigned orthology and the protein homolog tables, and then what we're seeing in the synteny views, it does look like that there are two analogs of the LUX gene in soybean and one in Phasolus corresponding to the Arabidopsis gene. And then you'll have to, well, you don't have to, but take my word for it that I actually did not look at this beforehand. But from this study, which actually involved transforming some of these genes and looking at the mutants, they did identify that the two homologous LUX gene pairs in the soybean genome are on chromosome 12, on chromosome 11, and they're precisely the ones that we identified in the orthology, and they're precisely the ones that we are seeing up here. So let me just, you see down here, it's 12 G06 0200, and the other one is, come on. The other one is 11G 136 600. So by combining just the protein sequence-based orthology and synteny, we get support for this identification of these homologs. Okay, so 2 copies of the LUX gene in soybean and 1 in Phasolus. How about ELF3, another component of the evening complex? So I am going to go back. Here, or do I want to show you? You know, I want to show you one more thing, since I know that people aren't always as comfortable using synteny and direct orthology calculations as they are blasting. So what would we find if we blasted? So let's take our Arabidopsis LUX gene. Let's go down to the peptide sequence, which is down here. It's the peptide sequence for the primary transcript. And we're going to blast it. But we're not going to blast it against Arabidopsis. We already know where it is in Arabidopsis. We'll blast it against soybean. Okay, and so this is a pure BLAST-P, protein set against protein set. You can see the top HSP hits here with links to their gene pages and their browser views. You can select them and add them to the cart if you wanted. There's also a graphic view, and there's also a standard NCBI BLAST view. One of the really useful things in this viewer is that these are all interlinked. So if I'm interested in this feature and I select it, it's selected in the table view and also in the NCBI text view, and those stay linked throughout. So I can, however I'm most comfortable interacting with the data in tabular form, graphical form, or in the NCBI format, I can do that. I can also download the full NCBI test set directly in whichever format's most useful for me. Anyway, so what do I see here? Okay, I see the top hits. These are different transcripts from the gene on chromosome 12 and one of the transcripts from the gene on chromosome 11. These are in fact from the two genes that we identified as being the analogs of the Arabidopsis lux gene. So we didn't see anything all that new here. However, if we're really paranoid, I can modify this BLAST job, and instead of searching the gene set in soybean, I can actually search against the genome, right? Maybe there's regions where for some reason a gene wasn't called, but there's very good hit there. So I'm going to do a tBLASTn BLAST alignment against the soybean genome. And that takes longer because the genome has to be translated. Okay. So now I'm actually not hitting genes, I'm hitting regions of the genome. What that means is there's no gene link anywhere. There's just a browser link to go to that region of the genome. And there's no ability to add stuff to the cart because all we add to the cart are genes and genes families, not genomic regions. That may actually change in a little bit, but right now, that's how it is. So let me get this out of the way. You can see the top hits are in the region of soybean chromosome 12 and chromosome 11, just like the genes we identified. Let's actually take a look at what's hitting. So I'm going to go to the genome browser here for soybean with this hit. Back out a little bit. Okay, and you can see that I'm hitting the coding sequence of the gene I identified before, 12G0, let's make sure, 12G060200. So no surprise, we're the best hit. Same thing over here. Let me look at where this guy is ending up. Okay, and it is lining up with the gene on chromosome 11 that I identified as the other homolog, 136600. So no particular surprises. There's a spurious splice introduced in the hit here, but that's BLAST for you. It's not a splice-aware aligner. So certainly what we're seeing in BLAST, both against the gene set and against the genome, is consistent with all the identifications we made before with synteny and orthology. So that's all good. Any questions at this point? So assuming I have time, let me just get started on finding that other component, the ELF3 gene. Okay, well, that was simple. So that's the only hit, and it's the one with this gene symbol assigned directly to it. And now I am going to look for soybean. There's a period. Right? And so again, pay attention to the score and the relationship and not the similarity. Even though this gene down here, right, has been assigned automatically that def line, it is not in the one-to-many relationship with Arabidopsis that we expect for the soybean gene. And then let me pull up another. Note here, which says for ELF3, there are, you can read this, 3 homologs of ELF3 and 2 homologs of ELF4 in soybean. So I do expect that these are the homologs. Now, is this also due to whole genome duplication and possibly a loss of one of them? I don't know, but we can certainly look at that. So let me go to the synteny view again. And this time I will try to work directly with an Arabidopsis synteny view initially. Actually, I'll save. Let me get the Phasolus analog. That'll be easier. I'll look in Phasolus again. It's just, since it's so much closer than soybean and it's one-to-one with the Arabi, it should have a stronger synteny signal. So Phasolus, soybean. Okay, so only 2 locations, 2 of the 3. Can you see the 2 green regions? So it's chromosome 1 in Phasolus, and it's chromosome 17 in soybean, and chromosome 14 in soybean. Those are the 2. The third gene, let me try to pull that right back. Where was the Arabidopsis one? It's not going to let me find it. I'll just search it out again. Sorry, guys. Should have. You said that those soybean genes were threefold. Okay, so 1 on chromosome 17, 1 on chromosome 14, and 1 on chromosome 4. So let me go back to that synteny view. Here's the one on 17. Here's the one on 14. I don't find one on chromosome 4, but actually, if you look in chromosome 1 Phasolus, to soybean chromosome 4, which is here, there is absolutely no synteny at all. Not with my gene of interest, not with any genes. So this is a case where there's been enough scrambling that any syntenic signal, if it was there, is not there any longer. So synteny can certainly support the association of the ELF3 gene in Arabidopsis and Phaseolus with these two examples in soybean, but not in fact for the third one. Even though the similarity signal and score signal is just as high, it is not in a syntenic region. So that's why it's useful to be able to combine a number of tools, not just rely on sequence-based orthology assignments and not just rely on synteny relationships, but actually to look at both. Okay, so the sequence-based orthology assignments certainly support there being 3 analogs of ELF3 within soybean. The synteny supports 2 of them, but not the third. And I'm going to add these 3 to my cart as well. And now I'm going to go to my cart and just quickly show you a few things you can do. Let's say you want to start analyzing the upstream regions from these genes and looking for promoters or such. So let me select them and click download. I'll focus on these specific transcripts. And that takes me into Biomart. And by default, it chooses sequences. In particular sequences, it chooses our peptide sequences. And just makes a FASTA file with gene name and transcript name as information in the headers. I can, however, choose to take the gene locus, or if I want the unspliced transcript, and include flanking region. So I could just say, I want to get the flank, the upstream flank from these genes, and I want whatever, 500 base pairs. Okay, and so this is this is 500 base pairs upstream of all of those genes. I could take that out and search in a promoter program if I wanted to. I can also say, you know what I'm interested for these genes? I'm interested in their orthologs, not just the ones I saw, but maybe in a bunch of other legumes. Right? Maybe I'm interested in them for chestnut and different soybeans than the particular strain I looked at. And cowpea. And that's going to be a very big set. Typically, and I would just say, just tell me when it's ready. Send me an email. I can work with the data offline. I'd be surprised if we get that before the end, but there it is. There's something in the chat. But I don't know. Anyway, that's one thing you can do in the cart. I can also save this list. Sorry, trackpad is a little jumpy. I won't bother with the description now. All right, and then now I have a list here that I can pull back in later, and I can also share it with someone. That's the sharing URL, and I can put that in the chat. And then everyone who's logged in should be able to get that and pull it into their cart. Okay, in the 45 seconds I have left, let me just show you the project. So, VMAP. This is the Brassicaceae map alignment project, looking at a bunch of, I think it's about 20 Brassicaceae genomes. So you can see the data set is restricted to those genomes off the bat. There's some additional information on the landing page that isn't there for the other projects. You can see that almost all of these are still restricted because they're still under analysis, but anyone can interact with the data. Brachypan is the Brachypodium pan-genome project, which you all know about. Now, these projects limit you to a particular set of genomes, but often you'll want to have an outgroup to work with. So that's why this, what we call the clade cut drawer here, is helpful, because even though the project is restricting you to these genomes, you can decide, well, like everyone else, the only thing I know is Arabi. So I want to include Arabi in everything I do. So now when I search, and now when I look at protein homolog tables and such, there'll always be an Arabi component within there. This is Tim Close's cowpea pangenome here with 8 strains. I think there's a manuscript being prepared on that. Epicon is a sorghum drought response project between UC Berkeley and JGI. Everyone at Hudson Alpha is familiar with the Gene Atlas project, which is looking at building gene expression resources across a broad swath of the plant phylogeny. Jim Laban's MAC Open Green Genome, trying to fill in gaps in the land plant phylogeny. And of course, Phytozome, which is everything. Okay,